Okay, good afternoon, everybody. It's a great pleasure to introduce Professor Samuel Berkovic today to speak about the genetics of human epilepsy. So Sam is a laureate professor at the Department of Medicine across the road at the University of Melbourne. He's a clinical neurologist and also a clinical researcher, and they've got very strong ties to basic research. And together with other collaborators in Adelaide and in Germany, they were the first people to discover one of the human epilepsy genes in 1995, and since then have continued to do amazing work finding many more epilepsy genes. And so this work has meant that he's been elected as a member of a fellow of the Academy of Science in Australia and also a fellow of the Royal Society in the UK. And it's really changed the way that people think about the causes of epilepsy, and therefore also the diagnosis of epilepsy and now how epilepsy may be treated. He's one of the co-investigators on an NIH-funded grant to be able to sequence 4,000 epilepsy genomes, and so you can imagine with this sort of work they'll find more and more genes that are involved in this disorder and understand more deeply about the genetics of human epilepsy. So please welcome him to give a talk today. Thank you.
Thank you very much, Marnie, and thank you for inviting me. Well, as you've just heard, I'm a clinician and really a clinical scientist, but the area where I work is in genetics, although I don't do the actual hard genetics. What I want to try and convey to you is how a clinician scientist tries to approach a disease, and then with partnering with the basic scientists, which the majority of you are, how that might work. And this is really essential in this area where translational medicine is so important. And sometimes when you look at clinical science and basic science, it's sort of like two solitudes, and that's wrong, and that doesn't lead to any progress. So I'll be talking to you largely on those on that sort of broad concept. I'm aware that my colleague Ingrid Scheffer spoke here a couple of weeks ago, so I'll try to not duplicate too much of what she said, although we work very closely together and much of what we do is shared. So in terms of understanding the causes of epilepsy, it's a relatively common condition, not nearly as common as diabetes or asthma, but it affects about 3% of the population at some time in life, and it's not one disease, it's many disorders. And we know that it's got both genetic and acquired causes. The acquired causes traditionally have been much better understood, things like epilepsy following head injury or following stroke or following perinatal injury or due to a tumour. And although genetics has kind of been there in the background, people are aware of the family history, it was only with the revolution in molecular genetics over the last couple of decades that we've been able to get real traction in understanding it. And in fact, as I'll show you, one of the things that has come out of this is that I think the data are now showing that genetics is actually far more important for this disease, or group of diseases, than had been previously thought. So there are many forms of epilepsy, and basically we think about them in two broad groups, what we call generalised and focal, and that refers to the pattern of seizures and the pattern of EEG that we see. The EEG is the electroencephalogram, and in generalised epilepsy the seizures begin bilaterally and simultaneously on both sides of the brain, whereas in focal epilepsy it begins in one particular part of the brain, often the temporal lobe or the frontal lobe, but anywhere from the cerebral cortex. Those focal epilepsies were relatively easy to understand in terms of acquired injury. You get a serious head injury, you're unconscious, you've got a hemorrhage in the brain, that area leads to epilepsy later on. But one of the surprising things that we found was that focal epilepsy could indeed be due to a genetic abnormality, and I'll be telling you about a new one today. These genes are everywhere in the brain, so why do they lead to a focal epilepsy? We still don't fully understand this. So some of you will have seen a convulsion, probably in the, at school or something like that, or in the street, but not everybody has. And just to give you a sense about, the burden that people with epilepsy have in their families, here's an example of a nasty generalised convulsion. It's a little bit asymmetrical. She's out of touch now. She's not in contact. If she were standing, she'd fall and possibly could, break a bone or get a secondary head injury. And there's this relatively prolonged tonic phase with the cry that you can hear. Patients can become incontinent at this phase. And these things in the individual, although they last only a minute or so, they're unpredictable. So the worry about having the seizure, about being seized, is with them all the time. And that's pretty scary and pretty spooky. It's a very dehumanising disease. When you see somebody with asthma who's breathless, you can kind of relate to that. We've all been breathless running up the stairs. But this sort of horrible thing where your humanity is taken away is pretty awful. And people have got, this has led to a lot of discrimination and a lot of difficulties dealing with the psychosocial aspects of it. This is reflected in this picture which hangs in St. Peter's Basilica. It's a painting by Raphael where the father in green is bringing his epileptic son to Jesus, who's out in the picture there, to be healed. And you can see that asymmetric tonic posture, a bit similar to what the actual seizure that I just showed you. And for this reason, epilepsy has been enveloped in being a spiritual disorder, and there's been a whole lot of nonsense and problem and issues about what its actual cause is through the years, and it's been associated with a lot of discrimination. It's actually been quite a difficult disorder to study scientifically for those reasons. And people want to know what's wrong, and we're pretty good at figuring that out. They also want to know, can we cure it? And in many cases we can control it, not always. And they also want to know what caused it. And one of the things that I think is very important as a physician, that I don't think in medicine we do very well, is that the identification of cause is actually really important. People know that shit happens. If you smoke and you get lung cancer, the patients with this, they kind of know, they accept it. But if you've got a beautiful child who suddenly starts getting this, and the doctor says, oh, I don't really know what caused it, that's incredibly unsatisfying. It's incredibly difficult for the families to deal with. They then go to the next doctor and the next doctor and then the witch doctor and the chiropractor and everybody else, and they're searching for because it actually is really important for closure. And I think we've underestimated the importance of this, and that's why what I hope I'll show you has become so important. So clinically, cause is very important. There's often a criticism about genetics that, you know, what's happened to gene therapy, you know, where is it? And certainly in neurological disease, it's almost nowhere. There are a few examples, of course, where there really is gene therapy. And whilst treatments coming out of genetics, I think, are going to be very important, almost as important for day-to-day management is this understanding of cause and explaining to people. And when they've got something they can hold on to, they can then, you know, look it up on the internet seriously, talk about it seriously, engage, and that actually helps. So what I'm going to do is tell you how I, as a clinician, look at the issue of a genetic contribution to epilepsy, talk about a selected number of relatively recent discoveries and the way we're thinking about it, and then say a few words about insights into mechanisms. And the impact of the new genetics is really, I think, just beginning, and it's a fantastic opportunity for the union of clinical and basic research. So, as I said, what caused it is a really important issue, and traditionally, and when I sort of was going through medical school and whatever, we thought that birth injury and minor falls might have had a lot to do with it. So, you know, many patients come with a view that or families had little Johnny not fallen out of the pram or not fallen out of the tree, or, as we'll discuss, not had the vaccination, they wouldn't have had epilepsy. And most of that's nonsense. However, I'm telling you that much of it's genetic. However, obvious Mendelian or recessive epilepsies are actually uncommon. So if you ask a physician who's not in the sort of epilepsy field, how often is epilepsy inherited? They say, oh, wouldn't think that was very common. But in fact, I'll show you that it probably is the major thing, and most patients don't have an obvious family history. So this is sort of the traditional pre-molecular genetic revolution view of the causes of epilepsy. This is based on reasonably solid epidemiology, that there are about a quarter of the causes that have an obvious acquired thing like stroke, trauma or congenital abnormality, and there's a large bunch that are, there's no obvious cause. So one of the ways I got into thinking about this was that there was a famous twin study by one of the founders of American epilepsy, a guy called William Lennox. And he studied twins, and twins are just a wonderful model for disease, because the classical paradigm, which was discovered by Francis Galton two centuries ago now, is that you compare the concordance, whether the disease occurs in the second twin after it occurs in the first twin, to identical or monozygotic twins, and compare that rate, the concordance, to that in dizygotic or non-identical twins who are genetically just the same as siblings. And here's one of Lennox's poster girls with their lovely 1950s outfits. And they had the onset of a mild epilepsy. Their intellect was normal, absence epilepsy or petit mal at the same age. And what happens with these twins, and we've got a large collection of these now, they start having seizures, often within days or weeks of each other, like a switch has gone on in the brain. It's quite remarkable. And here are the EEG patterns, the electroencephalogram. This little bit's normal, and this is what we call a spike and wave discharge. This is abnormal. During these periods, they are out of it. They're blanking out and not and unconscious. And twins are great subjects. They like being twins. We've got a wonderful twin registry here, and I've worked a lot with John Hopper, who runs a twin registry, to work on twins, as I'll show you. They're wonderful research subjects. These were studied in Boston, as you can see, in the late 40s. We actually found them, and you can see they like being twins still, as grandmothers. And they tell the story of complex disease, that is, disease that's non-Mendelian. They both had the disorder, so it screams at you that it's genetic. But none of their children and none of their grandchildren have epilepsy, and that's exactly what you see in a disorder that's due to multiple genetic contributions. So what I do as a clinical researcher is that I look at twin cohorts, and I use the twin cohorts, this experiment of nature. The basic scientists have their inbred strains. We use the twins and ask, well, you know, which ones are highly concordant? And this gives us extremely powerful information. So how do we go about looking at evidence for genetic contribution to disease as clinicians? Well, the first thing is to do what's called familial aggregation. We ask the question, are relatives of the patient in front of me more likely to have the condition than the man in the street? And we measure this by what's called the recurrence risk ratio. Where that's 1, it means there's no increased risk. It's nothing genetic at all. And the overall recurrence risk ratio for epilepsy is about 3. And for the major types, you can see it varies. For generalised epilepsy, it's about 6-fold. For focal epilepsy, that type where it's thought more often to be due to focal injuries, it's lower. And for febrile seizures with high temperature, which affect up to 3% of children, it's intermediate. And that fits with the clinician's view that most of the patients that he or she sees won't have an obvious family history. However, the story is very different with twins, and these are data from a study of about 400 twins that I've been doing in Melbourne. And what it shows, sorry about the typo here, what it shows is the concordance rate in MZs or identicals to DZs. And a concordance rate of 1 means that every pair that you see where one has it, the other has it. So a concordance rate of 0.8 is actually very high. And there aren't many diseases that go higher than this. So this screams at you that generalised epilepsy is a highly heritable condition, even though the recurrence risk ratio is here, and that's exactly what you would expect for a non-Mendelian complex disorder. What surprised us was that the rate in focal epilepsy was as high as it was, and in febrile seizures it was in the middle, and this is exactly the same trend as you see from the familial aggregation studies, giving us comfort that the twins are telling us the truth. So until the era of massively parallel sequencing, which we're in now, as you all know, the way we worked, and this was back in the 90s and early and last decade, was that we worked on these enormous pedigrees. As clinicians, we went and found, and we spent sometimes as much as a year constructing one of these large pedigrees. We then did linkage analysis. We figured out which segment was shared within the family, and then by positional cloning, as it was done then, found the gene. And this was the source of most of the gene discoveries now, and it's now got a lot easier with technologies where we can do genome-wide strategies. This old method is not yet dead, although we're using the new technologies better. And here's an example of something that has just appeared in Nature Genetics last month, and I think Ingrid may have mentioned it, but I think it's worth examining how this has really turned over our thinking about this. This is a family who seems to have an autosomal dominant trait, but the odd thing is that the colours of the individuals are different. The ones in red have seizures coming from the frontal lobe, the ones in blue have seizures coming from the temporal lobe, one had seizures coming from the frontal lobe, and there's a fair smattering of intellectual disability and autism in this family. And we didn't know which bits were actually part of the familial syndrome. We call this familial focal epilepsy with variable foci. In other words, the seizures come from different spots in the brain, which is very hard to understand, and we don't yet understand it. But we know what the gene is now. It was mapped to chromosome 22, and with massively parallel sequencing, the gene was found, and it's this gene, DEPDC5. We don't yet know what it does. It's a very large gene. It's perinuclear. It's responsible for essentially all the known families with this disorder, and there aren't many of them. But much more important is that we've started testing it in smaller SIB pairs or very small families, and it's responsible for about 10% of small families with focal epilepsy, which means it's probably the most important focal epilepsy gene so far found. So this strategy of sort of chasing families that tell us a story about genetics is still a productive one, and one that we're still continuing. So this gives sort of a schematic view about how we view epilepsy, and such a thing is sort of could be conceptualised for any complex disorder as opposed to sort of Mendelian disorders. On the y-axis is rough frequency, and we thought that there's a spectrum of epilepsies that are due wholly to genetic factors, and on the other end are the ones due to structural factors like trauma, strokes, etc., which, you know, we still know account for about 25% of cases. The early discoveries were sort of way over here, these sort of very highly Mendelian disorders that were solvable by the old technology, but we're now sort of moving into this area. But the DEPDC5 story tells us that, at least in some cases, the disorders that we find in extraordinary families that are telling us a specific story, the genes for those disorders may be relevant to the bulk of the cases here, and that's of course what we're currently trying to solve. So where we're at in 2013 with understanding epilepsies, and as a complex disorder, one could argue that it's one of the better understood ones, although there's still a lot to learn, is that many of the monogenic epilepsies, the ones on the left-hand side of that diagram, are due to ion channel abnormalities. And that makes sense. The epilepsy is a disorder of neurons. It's a hyperexcitable disorder, so something that regulates neuronal excitability makes a lot of sense. And the early discoveries involve both voltage-gated, that is, channels that change in response to sodium or potassium gradients, and ligand-gated, those that change in response to neurotransmitters like acetylcholine or GABA, and the first one that was mentioned was in fact an acetylcholine one. However, it's been increasingly clear that channelopathies are not the whole story, and a bunch of non-ion channel genes have been discovered, of which I'll be talking about Glut1, and I've already mentioned DEPDEC5. I won't go into the others. Similar to really the whole field of human genetics, it's become clear that disorders that the clinician thinks is unitary, as one disorder, are almost always genetically heterogeneous. In some ways this has been a disappointment, because we had hoped that if we thought about disease and could classify it clinically, then we'd have a list of genes and there'd be a nice one-to-one correspondence. And it's turned out that that's usually not the case. But that poses an even more interesting biological puzzle, because the answer then is, how do these multiple genes all presumably feed into the same networks and pathways to create the same phenotype? And that's a big problem for the next few years. And also there's pleiotropic expression of individual genes, which of course means that you have one gene and sometimes the same mutation that in the same family or in different individuals has quite different phenotypic expression. And the mechanism of that is also a challenge. In the complex epilepsies, the ones that we see most often in the community, where the recurrence risk is, down at 4 or 5, we believe that they are due to combinations of multiple genes or variants in multiple genes. And the big question here in all of complex genetics, not just in epilepsy or neurological disease, is whether this is due to combinations of common variants, which are detectable by GWAS or genome-wide association studies, of which there's sort of now about well over 1,000 in the literature. And whilst common variants do contribute to human disease, there remains a big debate as to whether it accounts for the majority of changes or whether it's just sort of a bit of icing on the cake. And we're currently involved in a consortium trying to sort this out in epilepsy. And secondly, and the hypothesis that I largely favour, is that, at least in epilepsy, it's more often due to rare variation. A relatively restricted number of genes in which spontaneous mutations occur. They have a lowered reproductive fitness, and you can't detect them by GWAS. You need to detect them by sequencing, and the modern age of massively parallel sequencing is the age where this will be solved, and the experiments are now being done and the results sort of just starting to trickle in. And as was mentioned, we're involved in this large NIH project to sequence 4,000 patients, and we hope we'll get a lot of answers from that. So that's what one might perceive from a sort of a classical clinical genetic view of this. But as they say, for the steak knives, there's more. And there's in fact more to genetics of epilepsy than what we can see from actually looking at patterns within families and in twins. First of all, we often don't get good family histories. If I asked you whether you knew whether your uncle had had seizures when he was a kid, you wouldn't know. It's not something you discuss at the dinner table, and it's not something that you get from a history when you see particularly a teenager coming into the clinic, and there's a lot that's missed. Secondly, an inobvious family history is just what you expect in a complex disorder, that it actually may not be there. But what we didn't expect was the importance of what's called de novo mutagenesis, that is new mutations. So patients with relatively common diseases having new mutations, and by the fact that they're new, you would not expect to see familial aggregation or even sometimes aggregation in twins, as we'll get to. And this has really changed the game in the severe childhood encephalopathies that Ingrid would have talked to you in detail about a couple of weeks ago, particularly one called Dravet syndrome, which I'll mention briefly. But it's also seen in milder diseases, and this has really been a big surprise. And of course, de novo mutagenesis is the usual mechanism in cancer, and we can take out the cancer and look for the new mutation, but it's much harder to do for a disease of the brain. But my bias is to believe that this is actually a more common mechanism, but that remains to be proven. So a few sort of vignettes about what we're doing and sort of what's hot. The first is Glut1, and this one was a big surprise. Glut, the reason you have your Wheaties in the morning and you feel awful if you don't, is that the brain runs on sugar. And Glut1 is the transporter that gets sugar into the brain, and without Glut1 you're in deep trouble. And we've known for about 20 years that there's a very severe childhood disorder with severe intellectual disability, small head, due to heterozygous Glut1 mutations. And you can measure the glucose concentration in the cerebrospinal fluid, and any sort of child neurology resident who missed that diagnosis would have a rather short career. But it's a rare disorder, and it's treated by the ketogenic diet, and these children are improved some by this. But that's what we thought was the story with the glucose transporter, and it's a fairly simple molecule. But a few years ago, it was found to be mutated in rather mild epilepsies and some patients who had what we call a paroxysmal dystonia. Dystonia is a twisting movement disorder which is believed to be mediated by the basal ganglia, deep structures in the brain rather than the cortex. So there were two structures in the brain that appeared vulnerable to this. And subsequently, we've been working extremely hard in looking at the full spectrum of Glut1 mutations, and I won't go through all the various phenotypes because that's something more for clinicians, but suffice to say it's turned out to be a far more common, it's a gene that's far more commonly mutated than we'd realised. And here's an example of one such small family where there are red and blue symbols, red representing the exercise-induced dystonia and blue representing a relatively mild epilepsy. Some patients have both, some have one or the other. And the important thing is that this is treatable by fairly simple dietary manipulation and doesn't necessarily need medication. So that was a really big surprise after we'd sort of been in this ion channel world. The second big surprise, which now a little old, but it's still very well worth knowing about. When I went through medical school and many others following me, we learnt that the DNA that we inherited from our parents was sort of invariant, a bit like the Bible. You couldn't change one letter or there was trouble. And if you did change one letter, it usually led to disease. So the big surprise came when the Sanger group, I think it was, had this paper in Nature saying, well, that's not actually true. There are large bits of DNA, what we call copy number variants, which typically encompass a number of genes that are either deleted or duplicated right through the genome. And this happens in all of us. We all carry these, and this illustration from that Nature paper is of what they found in normals and been amply validated. However, big ones, really big ones, have been found in a whole lot of neurodevelopmental disorders. And now this has become an essential investigation if we see a child with epilepsy and intellectual disability or epilepsy and autism. About 10% of them will have these copy number variants that are usually de novo and are basically the cause of their significant neurological disease. But more than that, rather than these sporadic children with nasty de novo mutations knocking out a bunch of genes, it's also emerged that copy number variation is a mechanism in complex disease. That is, it contributes but does not by far from completely explain that disorder. One of our previous fellows, when he went back to Germany, Ingo Helbig, reported that 1% of patients with a common type of epilepsy, idiopathic generalised epilepsy or genetic generalised epilepsy, which those twins have, the absence, 1% of them have a deletion at 15q that knocks out 6 genes. This deletion can be de novo or inherited, and here's a paradox of a severe mutation knocking out about 6 genes leading to a polygenic disorder. How do you explain that? Well, we haven't yet. There is one ion channel in that group, but we're not sure that that's the critical gene. And what's really interesting is that this explains to a small extent the comorbidity, in other words, the greater than expected co-occurrence of certain forms of epilepsy with autism, with intellectual disability and schizophrenia. It's not a high comorbidity, but those disorders also share an increased risk of these microdeletions. So that's been a thing that's really changed our thinking and the way we deal with patients, and there's a lot of very interesting biological questions to ask about how they might cause disease. Now, more or less our favourite gene up till DEP-DEC5 last month was the sodium channel, and the sodium channel is the critical or a critical channel for neuronal excitability. You'll probably remember from biochemistry that outside the cell the concentration of sodium is like 145 millimolar. Inside the cell it's like 10 millimolar. And that gradient's really important for monitoring neuronal excitability. So a lot of the energy we used is used for these channels to pump iron, literally. And there are a bunch of sodium channels in the brain, the most important of which is SCN1A. And Ingrid would have talked to you a little bit about this, and this is Dravet syndrome. And here the terrible story is of a kid who's normal up till 6 months, smiling, bubbling, happy, doing all the right things. Then he or she starts to get nasty convulsions. And in fact, the child that I showed you, or the young woman that I showed you in the first clip, had Dravet syndrome, but we didn't know that when we first saw her about 20 years ago, because this is all relatively new information. They may get status epilepticus, ongoing seizures associated with fever, and they get lots of other convulsions. And then in the second year of life, they get other forms of seizures, their development plateaus, and they're left, unfortunately, with chronic epilepsy, intellectual disability, and really very significant social disability. And they usually cannot live independently, with some exceptions. And the big discovery, which wasn't ours, it was made by a Belgian group, was that SCN1A is the key gene for this disorder, and about 75% of cases are due to this. And interestingly, there can be a combination of missense mutations, that is mutations that just change one amino acid, or mutations that lead to truncation of the protein. And most of them are de novo, that is, they are absent in the rest of the family. And the ones that aren't de novo remain an interesting mystery, but it's essentially a de novo disease. So this has completely changed the way we think about Dravet and related epileptic encephalopathies. That the term epileptic encephalopathies means where the seizures themselves are believed to lead to deterioration in brain health and intellect. And previously we used to see kids like this and we had no idea what the cause was. And now de novo mutations are clearly the major known cause. They can have copy number variations or single gene changes, and Dravet, as I've said, is responsible for about 75% of cases, and a variety of other related encephalopathies also have a pretty high hit rate. These are the first results from Epi4K, where we believe we've solved at least 15% of these, and I think many more will be solved as the analysis gets deeper. And Ingrid probably talked to you about her targeted gene approach that she's doing with the Seattle group, and they've solved a whole bunch of cases using that as well. And there's remarkable genetic heterogeneity. So we're thinking completely differently about these children. We're able to explain to the families the cause, and in some cases, so we've stopped the searching, and in some cases we've led to changes in treatment. Now I want to talk about an interrelated story that we think is one of the more important things that we've done over the years, and that relates to so-called vaccine encephalopathy. And this condition was alleged to be the development of encephalopathy with seizures and regression in normal infants following vaccination, and particularly the triple antigen vaccine was implicated, and this had major public health consequences where there were scares about pertussis vaccination. Families stopped having pertussis immunisation, and then pertussis increased, and pertussis is a lethal disease, a very unpleasant disease. It can be lethal, and you probably know there's been a minor epidemic recently as well in Australia, and it had medico-legal implications with very large awards and lots of nervousness about vaccination. Now, what do we know about vaccination and seizures? Well, vaccination can cause a fever. Those of you that have got children know that after their vaccines, they're often a bit grizzly and a bit grumpy, and they sometimes have a bit of temperature, and they get a bit of Panadol and everything's well. And we do actually know that seizures can occur with an increased frequency, and this is solidly shown by epidemiological studies, but it's benign. And multiple studies fail to show an association with encephalopathy. But parents want to know why, and the kid was normal before the vaccine, doctor, and look at him now. He can't talk, he can't walk like his brothers and sisters, and it happened after the vaccine, so ergo, propter hoc. Vaccination's blamed, and juries are sympathetic. So we, in the phase when we were collecting, we're still collecting, but when we were starting to really get to grips with this idea about de novo mutations and looking at Dravet syndrome, we had a collection of cases of of infantile encephalopathies which had an apparent vaccine-related onset. And the families believed that this was due to the vaccine, and many of the doctors believed that as well. Most of them had a diagnosis of Dravet syndrome when we looked at them in detail, and Dravet wasn't well recognised at that time, but when Ingrid and I went over the histories, there it was, they had Dravet syndrome. So we asked, well, do they have sodium channel mutations? And blow me down, they did. 11 out of 14 had mutations in SCN1A. And in the 9 cases where we could get DNA from the parents, they were de novo. So this explained the mystery, how this child coming from a blameless background in terms of epilepsy, no epilepsy in our family, how they could have a genetic disease, and they in fact had Dravet syndrome. And this got published in the Lancet, Lancet Neurology rather, and created a moderate amount of interest. I was still troubled by the issue that there were so many cases reported where the disorder seemed to start by after vaccination, and I developed a firm view that this was purely due to retrospective recall bias, and that we need to blame something. So Dravet starts at 6 months of age. What do you get at 6 months of age? You get your 6-month vaccination. And I expected that this was all just spurious recall. So how do you sort this out? Well, the best way is by a large prospective study, but Dravet is sufficiently rare that this would never be practical. It could be done, but it would be extraordinarily expensive and not really practical. So there's a wonderful research tool that most Aussie mums hang on to, and that's this, their Maternal and Child Health Centre book, which has all the formulas they're meant to give and the dates of their vaccination. And this happens to be my son Jonathan's book, and my wife and I we think, reasonably good parents, and he got his vaccinations, as you can see, stamped in there by Sister Fong, and the results seem to be really fairly satisfactory. He's 6 centimetres taller than me. So what we did was we got the vaccination books, because the medical records aren't terribly good on this, and matched that, the dates in those vaccination books, which are precise to within 24 hours, and matched it to when the first seizure was documented, which was generally well documented in medical records because the patients went to emergency departments, and at least until relatively recently, when hospitals chuck everything out after 7 years, you could get much of this. Another one of my hobby horses. So this graph shows the relationship of the first seizure to the vaccination beforehand. And 3 kids over here, and that there are, I think, 37 in all, 3 kids had not had a vaccination, so it could not have been related. But in all the others, they'd had their seizure after one or other vaccination. The colours refer to first, second or third. And what you can see immediately is that there is not a random distribution, but there is a peak within the first 48 hours after vaccination. That's the colour scheme of first, second or third vaccination, as it happened to be. So my hypothesis that this was due to spurious retrospective recall was incorrect. And in fact, this was sort of an added reason that was reinforcing the view, perhaps amongst doctors and certainly amongst families, that the vaccination had caused it. And of course, with the mutation there, and you never see these mutations in otherwise healthy children, it leads to the possibility that the vaccine sort of in some way triggered it. And the way to look at this a bit more carefully was to look at those children that had seizures at the time of the vaccination, and we call them vaccine proximate or vaccination proximate, compared to the rest. Did those groups differ? Were there 2 different diseases here? And the answer was that no, there wasn't. The only difference was that the vaccine proximate group did not differ from the rest, except that the onset was about 7.5 weeks earlier. So their age of onset was earlier. Their seizure types, their intellectual outcome, their degree of regression, all the other things we could measure was identical. And it seemed that the vaccine was just the event that brought this out, but it would have happened anyway because they had this nasty sodium channel mutation. But it to some extent explains the misconception about causation, we believe. So it appears to trigger onset, but is not a different phenotype, and explains a strong belief that the vaccination is causative. So we think that story is dead. Of course, the current issue with vaccination is autism, and the genetic causes of that are still being unravelled. But the case for the MMR vaccine causing autism is even weaker. So the next short story I want to tell you is again related to Dravet syndrome, and this sort of shows how a clinical observation may lead to understanding a biological principle. And here is a pair of twins with Dravet syndrome. They are discordant. They are monozygotic twins, and I can reassure the gentleman in the room that there are ways of changing the hair colour. And although they superficially look the same, they superficially look different, the twin with Dravet is a little chubbier. Look at the mouth and look at the nose and look at the eyes, and they're identical on molecular genetic testing. So this twin has Dravet syndrome. She has got a mild intellectual disability. Her co-twin does not have Dravet syndrome, is tertiary educated and, you know, an absolutely normal woman. So how can this be? So we ask, well, what might this be telling us? So the normal cause of Dravet syndrome is believed to be mutations in the gametes, more often in the sperm than the egg, but it can occur in either. Very occasionally we see healthy parents that give rise to more than one child with Dravet syndrome, and there we believe there is so-called gonadal mosaicism, that there's a clone of mutated cells in the ovary or in the testis, and sadly these parents then give rise to more than one affected child. There's also a phenomenon called somatic mosaicism, where in the case of Dravet syndrome, you see a child with Dravet syndrome, but their parent is much more mildly affected, normal intellect, just a few seizures, and what you see in them, and there are rather few cases of this that are discovered, is very low levels of the sodium channel, rather mutation, not the 50% that you'd expect in a heterozygote, but much less than this. And here we believe that the mutation occurred somewhere later in embryogenesis, such that the parent is a mosaic, but sadly one of the gamete that led to the birth of that child was unfortunately heterozygote, so the child is a full heterozygote, and that's somatic mosaicism. The twins fill in the story because one twin is a complete wild type, and we looked very hard for very low levels of the mutation, whereas the girl with Dravet syndrome is a complete heterozygote, and we looked in every tissue that we could find, and this implies that the mutation occurred in the very early embryo stage, probably at the 2-cell stage, one cell leading to a child with Dravet and one to a healthy child. So what this sort of story shows is that de novo mutations can occur at really any time in the cell cycle, and that got published in the New England Journal a couple of years ago. The story even gets more interesting, and this is not work of ours but of other groups. Here is a rather rare disorder called Hemimegalencephaly, and what that means is that half the brain's abnormal. This side is normal, but this side here has got altered patterning of the grey matter, this increased signal in the white matter. It's quite abnormal. These children are quite severely disabled. This disorder almost always, or essentially always, is sporadic, and the cause wasn't known. So it causes a severe and highly lateralised epilepsy, and in a couple of papers coming out last year, what they did was they compared tissue from the brain, because these children are operated on, and that was the sort of the advantage they had scientifically. They took tissue from the brain and compared it to the tissue, compared it to the DNA from leukocytes in those children, and what they were able to show was that there was a mutation in the brain, shown here, that was not present in the blood. So this was another example of a very late somatic mosaic mutation, presumably just affecting the neuroectoderm. Why just in one hemisphere and the other? Not completely clear, but leading to these, this quite remarkable disorder. So this disorder that we had no idea about its cause has been shown to be due to somatic mosaicism. And they found 3 genes, all nicely in the same pathway, so there's an evolving neurobiological story with this group. So we do know that post-zygotic mosaicism can cause mild epilepsies, like I told you with Dravet syndrome. It's important because of the recurrence risk in children that you need to, genetic counselling needs to be very carefully couched for these families. And is it a common phenomenon? Well, as I said, it's usual in cancer where tumours examined, and it may well underlie neurological disorders like epilepsy as well. So we don't know the numbers to this anymore to doctor what caused this. This fraction remains much the same. The only new thing that's been found is that there have been some immune causes of epilepsy, but my view is that much of the rest of this, the evidence is starting to point to much of it being genetic. So, again, that's a testable idea now, it's certainly not fact. So, how do these mutations cause these highly heterogeneous disorders? So here is a long story that I'll present briefly, but this is a family that we looked at in the late 90s with a very complex pattern of epilepsy. The individuals in red have childhood absence epilepsy, which is the type that I showed you in the twins, the little blinking out spells. And the ones in white, which are, as you'll see, the commonest, have febrile seizures, and there are a few others smattered in. And together with John Mulley's group and Robin Wallace, we mapped this family way back, and we found a mutation in the gamma-2 subunit of the GABA-A receptor, GABA being the major inhibitory transmitter in the brain. And the question remained, how can this mutation cause these highly heterogeneous phenotypes? And Chris Reed and Steve Petru across the road at the Florey have been working on this. And they have created a knock-in mouse with the same mutation, as well as a knockout mouse. In other words, a mouse with the identical missense mutation to the human family, and one where the gene is absent. And they're able to see absences, the minor seizures that these children have. They're minor but frequent. And there is the characteristic EEG, which is a little faster in mice than it is in man. And the interesting thing is that that absences are present in both the knockout, in other words, mice that only have one copy of the gene, and mice with the knock-in, with what we call the R43Q mutation. In the mice, it responds to a drug called ethosuximide, which is the drug of choice in patients, and it's therefore validated as a good model. And the inference then is that the absences are due to haploinsufficiency, that if you either you only have one copy of the gene, or you have one copy that's mutated and presumably non-functional, you get the same phenomenon, and the absences in those two strains is the same. However, in a different strain of mice, which is known to be more seizure resistant, we don't see absences, and therefore the absences are sensitive to genetic background, and that indeed would be the inference from the human patients as well, where the disorder wasn't completely penetrant in all individuals with the mutation. They also have a febrile seizure assay, and here they literally heat up the mice, and they look, using a survival curve plot, they look at when the mice have seizures, or the percent that have not had seizures. And the wild-type mice are shown in grey here, and what they're able to show is that mice with a much greater propensity to febrile seizures, the survival curve drops down really fast, is only seen in the knock-in. So the knockout mouse is not susceptible to febrile seizures. It has them at the same time or temperature as the wild-type. So that infers that the febrile seizure phenotype's a direct effect of the mutation, and is not sensitive to genetic background because it also occurs in the C57. So here is one mutation causing two apparent physiological mechanisms. Can we take that down any further? And here's the hypothesis for which there's some proof. So inhibition in the brain is of two broad types. There's phasic inhibition, which is like when you tap the brakes, versus tonic inhibition when you turn on the handbrake. And phasic inhibition occurs at the synapse. Tonic inhibition occurs extrasynaptically. And in these models, the physiology people working with Steve and Chris, although they're physiologists themselves, have shown that phasic inhibition is reduced in both, and tonic inhibition is abolished in the knock-in but not the knock-out. So here we've got a nice potential explanation. I don't think it's the complete explanation about how one mutation can cause this quite complex match of phenotypes in man that's been modelled in the mouse. So in conclusion, where we think this work is taking us is the role of epilepsy, genetics in epilepsy has been underestimated. Mendelian epilepsies remain relatively uncommon but are still a rich source of gene discovery, and we've worked on a number of rare Mendelian epilepsies with Melanie and Catherine Smith with great success, and that will continue, I believe. Familial aggregation is modest. De novo mutagenesis, I think, is emerging as really very important, has turned our thoughts about the importance of genetics on its head. We're slowly unravelling the genetic determinants of complex epilepsies. We think, but haven't really nailed yet, that the rare variant hypothesis may be more important than common SNPs, and that de novo mutagenesis, both with large effect genes and copy number variants, is going to be very important. Perhaps genetic factors explain much of the 70% of unknown causes, we don't know, but we're beginning now to find ways of understanding the very complex genotype-phenotype interactions. These are now tractable problems, I think, and they will lead, hopefully, to better treatments. So, as I said, I basically do the clinical side of this together with Ingrid and a number of our fellows. The molecular genetics has been done largely by our colleagues in Adelaide, particularly John Mulley, Leanne Dibbins and Joseph Getz and their labs. The functional studies over the road with Steve and Chris. We've had some very, very productive interactions with Melanie and Catherine here at the WEHI and a number of other international collaborations. Thank you very much.